Today we'll be talking about the applications of biotechnology. So until now we have learned how to use biotechnology, or rather gene editing or molecular biology techniques, to come up with a process or a product that is beneficial for us. So today we'll be talking about the applications of those processes or products, or how it is being applied to this day. We'll first talk about each of the industry that is there, like the agricultural biotechnological industry or the medical biotech industry, and we will be talking about each and every example within each of those industries. First off, with agricultural biotechnology, the crops which are produced using rDNA technology are known as GM crops, or genetically modified crops, or they can also be simply termed as GMO, which is a genetically modified organism, or they can also be termed as transgenic plants. Right? So any kind of modification that you do in any crop, for that matter, the end product of it would be called as a GM crop, which stands for genetically modified. So you have a genetically modified crop. It can also be simply termed as GMO, which stands for genetically modified organism, or the plants can also be known as transgenic plants. Now, why would you go through a GM crop? Why would you create a GM crop? There are multiple reasons why a GM crop can be created. One would be to obtain plants which are resistant to diseases, which are resistant to pests, which are resistant to environmental stresses. Environmental stresses can be anything like extreme cold, extreme heat, drought conditions, salinity conditions. So any of the environmental stress that can occur on a daily basis, creating a plant which is resistant to it. So creating a plant which can grow in low water conditions so that it can grow in a drought condition, or creating a plant which can grow in extremely cold conditions or extremely hot conditions. So that is why that would be environmental stresses. Reduction in seed loss. Reduction in seed loss specifically happens when a crop is being harvested. So after a crop has grown and you're going to be harvesting the seed, like rice or wheat or anything, there is some amount of loss of the seed occurring during the harvesting process. So a genetically modified crop can be programmed in such a way that the amount of seed loss that is occurring during harvesting will be reduced. Another thing that can be used is bioremediation. Bioremediation is when crops are being used to take out toxic substances from the soil. So if the soil is toxic for some purpose, for example, let's say that there is a high salt concentration in the soil and you want to get rid of the high salt concentration. So you would grow a specific plant, a GM crop, which takes out the high saline condition that is there in the soil. So bioremediation is when you are using plants to take out toxic substances from the soil. So you grow the plant in it, and while the plant is growing, it is going to remove all the toxic substances from the soil. GM crops are also designed for biofortification. Biofortification is when you are putting in a specific mineral or a vitamin or some sort of biomolecule into the plant, meaning that that plant is going to synthesize that biomolecule in excess, and that is known as biofortification. So if a population is lacking in a specific biomolecule, let's say a population lacks vitamin A, so you want to fortify that population with vitamin A, then you could just grow a crop which is producing excess of vitamin A, which is used as a staple diet by that population. For example, rice. So if you create rice which can synthesize excess amount of vitamin A, and that is something that is easily eaten by the majority of the population, then that leads to biofortification. So designing a crop that can synthesize a specific biomolecule in excess. It's known as biofortification. You can also create GM crops to produce medically important products. Medically important products can be anything from hormones, oral vaccines, or anticoagulants. So you can design a specific crop to produce a medically important product, so that whenever the plant is being harvested, you can also harvest or isolate out your medically important product that you wanted from it, like anticoagulants or oral vaccines or hormones. Some of the hormones can also be grown using GM crops. Another one would be elimination of allergens. Some people are allergic to certain crops, so some people have nut allergies, like they can't eat groundnuts and peanuts and stuff like that. So figuring out what is the allergic component within a product and then removing that allergic component by using a GM crop. So if someone has nut allergy and we can figure out what is the specific component within the nut that is causing the allergy, we just design the same plant without that specific component. So we remove the allergen and design a plant in that way. So elimination of allergen is one other application of GM crops. Improving shelf life is another major application of GM crops, so that any vegetable or fruit can be transported across vast distances without it degrading, so increasing the shelf life. So this also refers to ripening, so you reduce the ripening time, or rather you extend the ripening time, or you stop also the ripening of the fruit or the vegetable, by which you will be improving the shelf life of that fruit or vegetable. Modification of biomolecules is another application of GM crops. Modification of biomolecules refer to you are going to grow a crop in which a specific biomolecule is going to be enhanced. For example, if we are thinking about paper industry needs crops which have a lot of cellulose and starch in it. So if we can grow crops which are producing excess of cellulose, then it would be beneficial for the paper industry, and this will become something that is sustainable. So not many plants would have to be cut down to produce paper. You would just be using a fraction of those plants, because the plant that you have created, a GM crop that you have created, will be synthesizing or producing cellulose in excess. Okay, so modification of any sort of biomolecule which can be beneficial for a specific industry, so that it can be used, for example, paper industry, it can be used for detergents, it can be used for biodegradable plastics or things like that. So these are all the applications of GMOs or transgenic plants in case of agricultural biotechnology. So we'll be going through different examples for transgenic plants in case of agricultural biotechnology. The first example would be Bt cotton. Bt cotton is termed as so because the toxin that is put in, the gene for the toxin that is put in the cotton plant is from the bacterium Bacillus thuringiensis. The toxin that the Bacillus thuringiensis bacteria synthesizes is known as the delta toxin or the crytoxin. Now the toxin is specifically targeted towards bollworm. Bollworm is a cumulative term given for larvae, belonging to all of the three families, Lepidoptera, Coleoptera and Diptera families. And what happens with the delta toxin is that when the Bacillus thuringiensis bacteria is going for spore formation. So whenever the bacteria is going for spore formation, it is going to be synthesizing the toxin. But the toxin is in the inactive form, which is the crystalline form. Okay. So the toxin is always in the inactive form, which is the crystalline form. But whenever the larvae ingest the inactive form toxin, the toxin is undergoing proteolytic cleavage within the larvae. So it will undergo proteolytic cleavage within the larvae and it will become active. Whenever the delta toxin is active, it is going to attach itself to the epithelial cells in the gut of the larvae. Okay, so the bacteria is going to synthesize the toxin in an inactive form, which is a crystalline form, whenever the bacteria is going for spore formation. But when that inactive form toxin is being ingested by bollworm, which is any kind of larvae belonging to Lepidoptera, Coleoptera or Diptera, the crystalline form toxin is going to undergo proteolytic cleavage, wherein a part of the toxin, the amino acid chain, is going to get cut off. So when a part of the chain is cut off, the protein, the toxin in this case, becomes active. When the toxin is active, it is going to attach itself to the epithelial cells of the gut. It is going to attach itself to the epithelial cells of the gut, and it is going to create cationic channels in it. So there will be cationic channels which will be created in the plasma membrane of the epithelial cells within the gut of the larvae. Because of cationic channels being created in the plasma membrane of the epithelial cells are going to ooze out and die, and eventually that is going to spread all through the larvae, and the larvae is eventually going to die. So that is how the death of the larvae occurs, using the delta toxin or the crytoxin. Now what was done was to take the gene which codes for the delta toxin or the crytoxin from the bacterium Bacillus thuringiensis and insert it into a cotton plant. So the cotton is going to be the plant when it is growing, all of its foliage, meaning the green part of the cotton plant, on which the larva is going to be feeding. So the larva, how it grows is by feeding on the leaves of the cotton plant. So the toxin is going to be produced in the foliage portion of the cotton plant. So all of the leaves, the green part of the plant, is going to be producing the toxin. So whenever the larvae eats it, the crystalline form of the toxin is going to enter into the larva, and once it enters the larva, it is going to undergo proteolytic cleavage, become active, then it is going to attach itself to the epithelial cells within the gut, and once it attaches itself there, it is going to create cationic channels, and because of the creation of the cationic channels, the gut epithelium is going to die basically, and that will lead to the death of the larvae also. Now there are some uses, or rather advantages and disadvantages with BT cotton. Advantages being that of course it is effective against bollworm, so you are going to be killing off all of the pests that are there for the cotton plant, but it is not going to be killing out all of the pests. It cannot kill out pests which are succulent. So like I was saying, the delta toxin is going to be synthesized only in the green part of the plant, and it necessitates that the larvae eat, or any of the pests, it needs to eat the plant. Only when it eats the plant, the toxin enters the pest, and then it can kill the pest. Otherwise it is not going to kill the pests. So if there are pests which are succulent and not eating the plant, so there's a difference between succulent pests and pests which eat the plant. So larvae usually eat the plant, but succulent pests just attach themselves to the plant and suck the sap from the plant. So if that is happening, then the BT toxin is not going to be effective against those kind of pests. Another disadvantage of BT cotton is that it has destroyed pretty much all of the indigenous varieties of cotton plants which were there all over the world. Because BT cotton has been used so much all across the globe, indigenous varieties of cotton plants which were there in all of the countries have pretty much diminished in their numbers. Other than that, another disadvantage of BT cotton is that it is not going to be producing seeds. So the seeds that are there are not going to be fertile, so you cannot use the seed from the plant to plant another crop. You will need to buy seeds every single time from the company, and that is one more disadvantage of BT cotton. Another method that is used for pest control while creating transgenic plants is the RNAi or RNA interference technology. Now before we were talking about Bt cotton, wherein you're going to be taking a toxin from bacteria, Bacillus thuringiensis, and then inserting it into a plant so that it will synthesize that toxin, and whenever a pest is going to feed on the plant, that pest is going to die. So that is one method of pest control. Another method of pest control is RNAi. RNAi stands for RNA interference, and it makes use of two different kinds of RNA, miRNA and siRNA. miRNA is microRNA. It is anywhere between 20 to 25 nucleotides in length, the RNA itself, but the active form of the RNA, the one that takes part in RNAi, is usually 22 nucleotides in length. It is specific to mRNA only. It does not digest out any other kind of RNA other than mRNA. It is conserved, meaning that the sequence of miRNA, the microRNA, is conserved all across the kingdoms. So any organism that you take, the miRNA sequence is pretty much conserved. It is derived from single strand RNA that folds back on itself to create a hairpin loop. So miRNA is a single strand RNA molecule. So you have a single strand RNA molecule, and this RNA molecule is going to fold back on itself in this way, and it is going to form base complementarity bonds. So this single strand RNA is of course going to have nucleotides like A, T, G, C, T, T, A, etc. Now when this RNA is going to fold back on itself, on the other end, if there are bases that are complementary to this, meaning that if there is a T here, that will be complementary to the A here, and same goes for A here, which will be complementary to the T here, a C which would be complementary to the G here, a G which would be complementary to the C here. So this is going to turn on itself and form bonds here, and therefore you would end up with a double strand looking miRNA. Okay, so miRNA is originating from single strand RNA, a single strand RNA which is going to fold back on itself to create what we call a hairpin loop. Okay, that is where the miRNA originates from. As for the siRNA, which is small interference RNA, it's around 20 to 25 nucleotides in length, meaning the active form of the siRNA, meaning the siRNA that is going to take part in the RNAi pathway is going to be around 20 to 25 nucleotides in length. It originates from double strand RNA, which are going to get cut into small pieces of 20 to 25 nucleotides. Okay, now moving on to the RNAi itself. RNAi makes use of a complex known as the RNA-induced silencing complex, termed as RISC. Now within the RISC complex there are multiple enzymes which are going to be active, but we will be only talking about two enzymes, dicer and the argonaut. So whenever miRNA or a siRNA is going to come in contact with the dicer, first off, why would miRNA or siRNA be deployed? It is when the cell feels that it is being attacked. So RNAi is a sort of immune response, meaning that whenever the cell is going to get some sort of a foreign DNA or RNA into it, it is going to deploy the RISC complex to digest out that RNA and DNA. Okay, so how are we going to use it to create a transgenic plant? The way we are going to use it is that whenever a pest is going to be eating the plant, the transgenic plant in which the RNAi is active, the miRNA and the siRNA are going to get into the pest. And in the pest within itself, the miRNA and the siRNA are going to become active. They will use the RISC complex and then break down the RNA which is there within the cells of the pest, essentially killing the cells, and then by prolongation killing the pest itself. Okay, so that is how RNAi will be used. So going back, so whenever miRNA or siRNA are there are two enzymes that we will be basically talking about. RISC complex has multiple enzymes, but we will only be focusing on dicer and argonaute. So what happens with dicer and argonaute is that when miRNA or siRNA are there in the cytoplasm of the cell, the argonaute is going to come and attach to the miRNA or the siRNA. The dicer molecule is going to cut the mi or the siRNA. So miRNA is created from a single strand RNA which is going to loop on itself, so it's going to look like a double strand RNA. siRNA originates from double stranded RNA, so it's any which way is double stranded RNA. So you have double stranded RNA to start off with. At this point comes dicer, and dicer is going to cut the double strand RNA so that each portion of the cut is going to be anywhere between 20 to 25 nucleotides in length. Okay, so it's going to take the long miRNA or the long siRNA, and then it's going to cut it into pieces which are approximately 20 to 25 nucleotides in length. Now the argonaute, which is bound to this complex, the double-stranded miRNA or the siRNA, which is approximately 20 to 25 nucleotides in length, the argonaute is going to bind to it, and when it binds to it, it is going to select one of the strands. So let's say that this is the argonaute protein and it is binding to the double-stranded RNA molecule, which can be the mi or the siRNA. This argonaute protein is going to select one strand, okay, and the other strand is going to be released out. So it's a double-stranded RNA molecule. So the bonds between the nucleotides are going to be broken down by the argonaute protein, and it is going to release one of the strands. So you will be left with only one strand of either the siRNA or the miRNA, which is 20 to 25 nucleotides in length. Now this strand of mi or siRNA is termed as the guide strand. Okay, so the argonaute, which is a complex of the RISC, so it is a part of the RISC complex. This argonaute is going to go find a mRNA or any sort of RNA that is there in the cell. With respect to miRNA, it is specific to mRNA, but it does not need complete complementarity of the mRNA. With siRNA, it is not specific to only mRNA, it can degrade any kind of RNA, but it requires complete complementarity. What is complete complementarity? Complete complementarity means that its target RNA needs to have complete base pairing with the guide RNA. So the guide strand of the RNA, which is currently attached to the argonaute, if it is going to have complete complementarity to the target, this occurs only in case of siRNA. In case of miRNA, it does not require complete complementarity. Okay, so whenever the guide strand, which is attached to the argonaute, comes across any sort of RNA, it can be mRNA, rRNA, tRNA, any kind of RNA, if there is complete complementarity, in case of siRNA, the dicer is going to again cut the RNA. Okay, so that is how the RNA gets degraded in case of siRNA. In case of miRNA, it does not require complete complementarity. So even if a few bases are complementary, then the RISC complex is going to cut this RNA, the target RNA, into pieces. So the RNA of the target cell, which in this case would be a pest, would be completely degraded. So any kind of RNA that is there in the pest cell is going to be completely degraded, and if RNA are not there, then any sort of protein will not be synthesized, and if proteins are not there, the cell cannot function, so it will eventually die. So that is how the death of a pest occurs if you are using RNAi. So just to recap, RNAi is RNA interference. It makes use of two different kinds of RNA, miRNA, which is microRNA, or siRNA, which is small interference RNA. In case of miRNA, its active form will be around 22 nucleotides in length. It is specific to mRNA, it is conserved all across the systems, and it originates from a single strand RNA molecule which is going to fold on itself and form a hairpin loop. In case of siRNA, its active form is around 20 to 25 nucleotides in length, and it originates from double strand RNA molecule. miRNA is specific to mRNA only, meaning that its target is only mRNA. In case of siRNA, it does not have a specific target, it targets every kind of RNA that is there. For miRNA, it does not require complete complementarity. So between the miRNA and its target mRNA, if there are a few base pairs which are complementary, the risk is going to cut that mRNA to pieces. With siRNA, it needs complete complementarity with its target RNA. So any kind of RNA that it is targeting, it requires complete complementarity. So the siRNA needs to have complete base pairing with its target RNA, and only then the RISC complex is going to cut it into pieces. So whenever mRNA in case of miRNA and any kind of RNA in case of siRNA are going to be cut into pieces, then they are not functional. If RNA are not functional, then you don't get proteins, you don't get any sort of functional components within the cell, because rRNA, for example, make a functional component of ribosomes. So if you don't have any sort of RNA within a cell, a lot of functions are not going to occur, which eventually leads to the death of the cell. And once cells start dying, it eventually leads to the death of the pest. So this is how RNAi can be used as a method for pest control in case of transgenic. Another example of transgenic plants would be herbicide resistant plants. So herbicides are usually used in case of any sort of agricultural practice to get rid of weeds. So herbicides are used for that purpose. The problem with herbicides is that a herbicide is not going to select which kind of plant it is going to kill. So herbicides are chemicals at the end of the day, so they are going to kill anything that they are potent against. So if you're going to be using herbicides, you need to make sure that your crop is resistant to that herbicide. Most of the times that is not the case, so it becomes a balancing act between how much herbicide to use to make sure that you kill the weed but also grow your plant, which is not an easy thing to do. So the example that we will be taking here is the herbicide glyphosate. It is effective against plants, or rather weeds, which have broad leaves, and it is effective against grasses. The way it functions is that it is going to stop protein synthesis in the cells, and it is going to stop protein synthesis by stopping the synthesis of 3 amino acids: tryptophan, tyrosine, and phenylalanine. So because these 3 amino acid synthesis is stopped, a functional protein cannot be synthesized within the cell. If proteins are not there, then the cell eventually dies out, and that eventually leads to the death of the weed, in which case it is broadleaf weeds and grasses in this case. Now the agricultural biotech application in this case was to synthesize, or rather create, a transgenic plant which would be effective, which would be stopping the function of glyphosate in itself, meaning that the plant will not be any more susceptible to the herbicide glyphosate. So even if you put glyphosate onto the plant, it is not going to kill the plant. How it was done was that there was an enzyme extracted from the strain CP4 of Agrobacterium. So CP4 strain of Agrobacterium synthesizes an enzyme which actually stops the function of the glyphosate herbicide. So the CP4 strain of Agrobacterium synthesizes an enzyme which stops the action of the glyphosate herbicide in the plants that it contains. So this enzyme coding gene was put into plant, and whichever plant was synthesizing this enzyme was resistant against the herbicide glyphosate. The first resistant plant which was created was soybean, and that was done in the year 1996. So to recap, glyphosate is effective against broadleaf weeds and grasses. It stops protein synthesis by stopping the synthesis of three amino acids, tyrosine, tryptophan and phenylalanine. Now how a herbicide resistant transgenic plant was created was by extracting an enzyme from Agrobacterium strain CP4, and this stops the function of glyphosate in case of those plants which have this enzyme. So the gene which codes for this enzyme was put into the crop, creating a transgenic plant. So whenever glyphosate was sprayed on these plants, it did not kill the plant. The first resistant plant was soybean, which was created in the year 1996. The next application or the example that we'll be talking about is a transgenic plant which has an increased shelf life for its produce. So it can be a fruit or a vegetable which has an increased shelf life, which means that it has slowed down or reduced ripening. Okay, the enzyme that we will be talking about is the Paul galacturonase. Now Paul galacturonase has one function, which is to break down pectin. Now pectin is a very big component of the cell wall of the outermost covering of either fruits or vegetables. So the outermost covering of any of the fruits or vegetables is made up of a higher concentration or a higher level of pectin in combination with cellulose and hemicellulose, of course, but it has a higher level of pectin. And the function of the enzyme Paul galacturonase is to break down pectin. So whenever pectin is broken down, the outermost covering of any of the fruit or the vegetable becomes more susceptible to infections, especially fungal infections. So things like tomatoes or fruits or any of those things that you take, when the outermost covering of the fruit, tomato for that matter, becomes active, the Paul galacturonase is active in them, then the outermost covering is becoming more susceptible, and therefore it becomes a very good environment for the growth of any sort of bacteria, or mostly it's fungi which grow on them, which we do not want because we want to increase the shelf life of the product. So the only way to do it would be to stop the action of the enzyme Paul galacturonase so that it is not going to break down pectin, which means that the outermost covering of any of the fruit or the vegetable is going to remain intact. If the outermost covering is going to remain intact, then it is not susceptible to infection, so the fruit is not going to get destroyed because of a fungi or a bacterial infection. The first transgenic plant which was created was Flavor Saver, which is a tomato variety. What they did was to use the antisense RNA technology in this case. Now antisense RNA is very much different from the RNAi tech. RNAi is RNA interference, and that uses microRNA or small interference RNA, miRNA or siRNA. Antisense RNA tech is going to use, or something that we call as RNA silencing also. Antisense RNA tech is also known as RNA silencing, in which case we use something known as an antisense RNA. So antisense RNA is a mRNA, is an RNA molecule which is going to be completely complementary to the RNA molecule of your target. So the thing that we are targeting here is the enzyme Paul galacturonase. Because it is an enzyme, it needs to undergo transcription and translation for a functional enzyme to come. So for translation to occur, you need a mRNA. So we have a mRNA which is going to be coding for the enzyme Paul galacturonase. So here is your mRNA which is coding for the enzyme Paul galacturonase. Now we are going to create an antisense RNA which is going to be completely complementary to this mRNA. So your antisense RNA is going to be completely complementary to the mRNA molecule, which means that it is going to go for bond formation. So the nucleotides which are there on the mRNA are going to form bonds with the nucleotides which are there on the antisense RNA. So you are going to end up with a double-stranded RNA molecule instead of a single-stranded RNA molecule. So what is going to happen? Because it is a double-stranded RNA molecule, this cannot be recognized by the ribosome complex. So for translation initiation to occur, the 16S rRNA which is there in the ribosome complex needs to form a complementary bond with the mRNA molecule here. Only then translation initiation can take place. But because this is a double-stranded molecule here, the 16S rRNA which is there in the ribosome cannot attach to this mRNA anymore, because there is no free site. The antisense RNA is completely covering up that site. Okay, so translation will not take place. If translation is not going to take place, you will not have the enzyme Paul galacturonase. That is how RNA silencing or antisense RNA tech works. Now, because this is double-stranded, it can also trigger the risk complex, which we have already spoken about. It is not that it always triggers the risk complex, but once in a while it can also trigger the risk complex. So RNA silencing majorly works by the fact that the mRNA is not going to be available for it to be attached to ribosome and for translation to occur. Because antisense RNA is attached to the mRNA, it forms a double-stranded RNA instead of a single-stranded RNA, and therefore translation is not going to occur. So your gene will not be synthesized. Not the gene, but the enzyme will not be synthesized anymore. But because this is a double-stranded RNA molecule, it can also initiate the risk complex, wherein the risk complex is going to come and attach to it and then break it down into pieces, like it would do either the miRNA or the siRNA. Okay, but this is not a normal occurrence. It happens once in a while. Majority of the stopping happens through antisense RNA tech, wherein translation is stopped. Okay. Another application with transgenic plant would be the synthesis of hirudin. So when I was talking about agricultural biotechnology and its applications, we were talking about creating transgenic plants which can be used to synthesize medically important products. So hirudin is one of those medically important products because hirudin is an anticoagulant. It is originally found in the salivary glands or the saliva of leeches, and hence the name hirudin, because it is found in leeches. Now it is synthesized as a complex with oleosin protein in any oil producing, specifically seed oil producing plants. Oleosin is a structural protein of oil bodies, or lipid droplets. If you think of mammals store fats in the form of adipose tissue. So adipose tissue is the storage for fats in case of mammals. For plants, the storage of fat occurs in spherical structures known as oil bodies or lipid droplets. So you have oil bodies or lipid droplets, and these are spherical structures within the cells which are going to be storing fat. The kind of fat that they store is TAG, which is triacyl glycerol. Now oleosin is a structural protein, meaning that it is found on the surface of the lipid droplets or the oil bodies. Okay. Now lipid droplets or oil bodies of course will be found in excess in seed oil. So seeds which are going to be producing oil like groundnuts, sunflower, rapeseed in this example will be having a higher concentration of lipid droplets because they are going to be accumulating fat, of course, which is going to give rise to oil at the end of the day. Now every single lipid droplet is going to have a structural protein on its surface, and these structural proteins are known as oleosin proteins. So hirudin is going to be attached to oleosin protein. So every oleosin protein is going to have hirudin attached to it. So how is the transgenic plant synthesized? You are going to take the hirudin gene, so the gene which synthesizes or which gives rise to the hirudin molecule is going to be attached to the oleosin protein gene. Okay, so you are going to have the oleosin gene, which is usually found in any plant that is going to produce seed oil. So you are going to take the hirudin gene, the gene which gives rise to hirudin molecule, and then attach it to the oleosin gene. So whenever oleosin is being synthesized in a plant, specifically seed oil producing plant, where it is synthesized a lot because it's a structural protein for the lipid droplet or the oil body, with it connected to it, hirudin is also synthesized. So you would have the polypeptide which is going to give rise to the oleosin, and attached to it will be the polypeptide which is going to give rise to the hirudin molecule. So whenever oleosin is expressed, at the same time hirudin is also expressed as a chain. So because oleosin is a structural component in case of lipid droplets, hirudin molecule is also attached to it. How are you going to take it out from the transgenic plant? Whenever seed oil is being extracted from any of these plants, so if you're going to extract rapeseed oil or groundnut oil or palm oil or sunflower oil, any oil producing plant to which you have attached the hirudin gene, whenever you extract out the oil from these plants, you're going to use a endopeptidase, a specific endopeptidase, which is going to break the bond between the oleosin and the hirudin. So the hirudin is going to be freed from the oleosin molecule. So this hirudin which is attached to the oleosin will become free, and these hirudin molecules can be isolated, purified, and used as an anticoagulant further on. So that is how hirudin can be used or rather produced using a transgenic plant. The next example that we'll be talking about is golden rice. Golden rice is a very good example for biofortification. Biofortification is when you are creating a transgenic plant that can synthesize a specific biomolecule in excess, so that a specific population which has a deficiency of this biomolecule can be fortified. So for this example, golden rice is going to be synthesizing in excess beta carotene. Beta carotene is the primary dietary source of provitamin A, which after the example that we'll be talking about here is golden rice. Golden rice is going to be synthesizing beta carotene in excess. Beta carotene is the major dietary supplement or the source for provitamin A. Provitamin A, after further processing, becomes vitamin A, which is a very necessary dietary component in case of human beings. If it is not there, it is going to lead to certain diseases. So if a population is there which is lacking vitamin A or has vitamin A deficiency, all that they would need to do would be to consume golden rice, so that they would get in excess beta carotene, which is essentially the primary dietary source for provitamin A. So what they did was to take the gene that codes for beta carotene and then insert it into the genome of the rice, hence creating beta carotene, hence creating golden rice, and it is termed as golden rice because beta carotene has a yellowish yellowish color to it, and therefore rice which is going to be synthesizing excess of beta carotene is going to have a yellowish color to it, and hence it was termed as golden rice. So that is another example for transgenic plants, and this one is specifically an example for biofortification. Moving on with applications of biotechnology in the medical field. So medical biotechnological applications is what we'll be talking about today. We'll be going with examples of insulin and monoclonal antibodies. Now moving on with insulin, the primary structure of bovine insulin was given by Frederick Sanger in the year 1951. Insulin until now has been either extracted from pig, so it's porcine insulin, or from cows, then it is known as bovine insulin. Now porcine insulin is the most similar one to human insulin. It differs from human insulin with just one amino acid. So the difference between the human insulin and pig insulin, which is porcine insulin, is just one amino acid. The difference between human insulin and cow insulin, the bovine insulin, is 3 amino acids. So pig insulin, the porcine insulin, is the one that is most similar to the human insulin. The bovine insulin is also used because the difference between human insulin and bovine insulin is just 3 amino acids. So before medical biotechnological applications, insulin was extracted either from pigs or from cows and given as a supplement for those suffering from diabetes who could not synthesize insulin on their own. Insulin is synthesized in the beta cells of the islets of Langerhans. It is made up of 51 amino acids total. It is a dimer. Contains one A chain and one B chain, and these two are connected to each other with a disulfide linkage. There are 2 disulfide linkages between the A chain and the B chain. Now before we get into how the medical biotechnology actually happened, we'll be talking a little about how insulin itself gets synthesized. Now insulin itself of course gets synthesized in the beta cells of islets of Langerhans. It is synthesized as a pre-proinsulin which undergoes several processes before it becomes proinsulin. Proinsulin is present in the Golgi apparatus, and their proinsulin is actually made up of 3 subunits. You have A, B and C subunits. All of them are connected with a total of 3 disulfide linkages. So you have a total of 3 disulfide linkages which are going to be connecting A, B and C subunits of the proinsulin. Now, if you want to get insulin from the proinsulin, you will need to remove the C subunit. So removing the C subunit, you will end up with just the A and B subunit. These two are connected to each other by 2 disulfide linkages, and this will be the active form of insulin. Now this is what you want to get out of a transgenic animal or a transgenic microorganism that you will be using for the synthesis of insulin. For the synthesis of insulin, microorganisms are used, not animals, and the vector that is used is PUC18 or PUC19. PUC18 and PUC19 vectors are pretty similar to each other, the only difference being that the multiple cloning site between PUC18 and PUC19 is just reversed. So it's in the opposite direction in case of PUC19. Now PUC18 or 19 of course have 2 selectable markers. They have an ampicillin resistance gene and a LAC-Z gene which is going to be coding for the enzyme beta galactosidase. Now what was done was to connect the beta galactosidase gene on the PUC18 or 19 with the gene that is going to be synthesizing insulin molecule. But because insulin has 2 subunits, A subunit and B subunit, which need to be connected through a disulfide linkage, this does not happen in prokaryotic organisms. Disulfide bond formation is not going to happen in prokaryotic organisms, so this needs to be done outside of the bacterial cells. Therefore, the thing that was done was to take one PUC18 plasmid, and you have the LAC-Z gene here, to which is attached the gene which is going to be coding for the A subunit. Okay. This is one PUC18. So you have one PUC18. Let's say that this is A, which is connected to, here you have the LAC-Z gene, and this will be the A subunit of the insulin molecule, and then you have another plasmid which again has the LAC-Z gene. So let's say that this is PUC18B, and this is attached to subunit B, and this will be LAC-Z gene. So this is the B gene, this one is the A gene, and both of them have the LAC-Z gene. So this is PUC18, one PUC18 plasmid in which the LAC-Z gene is attached to the A gene, the one that codes for the A subunit of insulin molecule, and another PUC18 which has LAC-Z gene which is attached to the B gene, which is going to be coding for the B subunit of the insulin molecule. Both of these are going to be put into different bacterial cell. You have one bacterial cell which would be having PUC18B, and another bacterial cell which is going to be having PUC18A. So these two plasmids are going to be put into two different bacterial cells. These two bacterial cells are going to be grown separately. Of course, PUC18, PUC19 has a very high copy number, anywhere between 500 to 700. So each bacterial cell is going to contain 500 to 700 copies of the plasmid, and each plasmid is going to give rise to the gene product, which will be a fusion protein containing beta-galactosidase attached to either the B subunit or beta-galactosidase which is attached to the A subunit. So this bacterial cell is going to give rise to a fusion protein which will be beta-galactosidase attached to the B subunit, and this is going to give rise to beta-galactosidase which is attached to the A subunit. So this will be a fusion protein, something like what was done with hirudin, where hirudin was attached to the oleosin protein. So you will end up with a fusion protein where beta-galactosidase is attached to the B subunit in this bacterial cell, and in the other bacterial cell you will have the beta-galactosidase enzyme attached to the A subunit. These two are going to be isolated from the two bacterial cells separately. Cultures, of course, you will not be using one single bacterial cell. So this culture, from this culture you are going to isolate beta-galactosidase attached to the B subunit protein. You are going to isolate it, and from this culture you are going to take out beta-galactosidase which is attached to the A subunit. Of course, when it is isolated out, you are going to use a specific endopeptidase, the same way that you would use in case of the hirudin molecule. So you are going to use the endopeptidase specific for the breakdown of the bond between the beta-galactosidase and the B subunit, and the beta-galactosidase and the A subunit. So you will have free A subunit and free B subunit. So these two are going to be isolated out, and the thiol group which is there in the cysteine molecules on the B subunit and the A subunit will be activated. When the thiol group is activated, a disulfide bridge can be formed. So you have one cysteine molecule which has a thiol group, another cysteine molecule which has a thiol group. Whenever the cysteine molecules thiol groups are activated, they can form a disulfide bridge. In between the A and B subunit, there are two disulfide bridges. So two cysteine molecules in each of them, the thiol group will be activated and two disulfide bridges will be formed. When two disulfide bridges are formed, it becomes an active insulin molecule, and this can be injected into a patient who is suffering from diabetes. This is how insulin is harvested from genetically modified organisms. Another application of biotechnology in case of medical biotechnology is the synthesis of monoclonal antibodies. Monoclonal antibodies are those antibodies which are originating from a single parent cell, meaning that all of the antibodies are originating from one single cell, hence the name monoclonal. All of them are clones of one single thing, one single cell, so they are all originating from a single parent cell, and also they all recognize only one epitope from the antigen. So interaction between an antibody and an antigen is between the antigen's epitope and the antibody's paratope. So the paratope which is there on the antibody is going to attach itself to the antigen's epitope. So the epitope and the paratope are going to attach to each other, and this is a sort of a lock and key mechanism that is extremely specific. So one epitope will always be recognized by only one antibody, and that is monoclonal antibodies can recognize one single epitope. If it is a polyclonal antibody, then it can recognize multiple epitopes. That is not what we are going to get into. Now, Georges Kohler and Cesar Milstein in the year 1975 were the ones who came up with the hybridoma protocol, which we will be discussing further. There were some problems with the hybridoma protocol, so it is not currently used right now. It was Greg Winter who came up with more humanized antibodies in the year 1988. Humanized antibodies meaning that they are not going to bring about any sort of immune reaction in the people who are taking these antibodies, because hybridoma technique uses antibodies which are initially extracted from any sort of animal. So any sort of animal like rabbits, rats, mice, goat and horses are the ones that are usually used as the parent animal from which the antigen and the antibody is extracted. So because it's from an animal source, there were some people who would have an immune response to that antibody, but that is the reason why the hybridoma technique is not currently used for the synthesis of antibodies which will be used for human purposes. There comes Greg Winter's humanized antibodies which occurred in the year 1988. Now going with the hybridoma technique itself, before we get into the hybridoma technique, just a quick recap of what happens with antibodies or which of the cells generally in the human system produce antibodies. Now antibodies are generally produced by the B cells. So the B cells are the ones that are going to be producing antibodies. So you have B cells. B cells are the ones that are going to be producing antibodies in case of humans. Now B cells are a class of lymphocytes. They are also known as B lymphocytes, which is a class of white blood cells. B cells have something called as BCRs on them, which are B cell receptors. So these are the paratopes onto which the epitope of an antigen can come and attach to itself. So every B cell is going to have a receptor on top of its plasma membrane or cell membrane to which an antigen can come and attach itself. And this is very specific to B cells. This attachment of antigen happens in the spleen, and this is where the B cells get activated. The B cells are going to be synthesized, or the B cells are going to originate from the bone marrow in case of human beings. That is where they originate from, the bone marrow in case of human beings only, and they get activated in the spleen where the maximum amount of antigens come and accumulate. So in the lymphoid system is where the antigens accumulate the most, and spleen is a part of the lymphoid system. The lymphoid system is majorly made up of the spleen and the lymph nodes. So the B cells, once they originate and mature from the bone marrow, they are going to move to the spleen, and in the spleen the B cell receptors are going to get attached to the antigen, and that is where the B cell gets activated, and then an immune response starts off. That is not what we are going to get into. Just remember that the spleen is where the B cell gets activated, and it is also the place where the B cell attaches itself to an antigen and gets activated. So this is what we are going to make use of for hybridoma technique. Now in case of hybridoma technique, any animal you can take. So here I am going to be talking about rabbit, but you can also, the other kinds of animals that are used are rats, mice, goats and horses. So these are the usual animals that are used for the hybridoma technique. So in this example I will be going with rabbit. So a rabbit is injected with an antigen for which you want to create an antibody. So let's say there is a specific antigen, for example an antigen that is originating from pollen grains. So if there is a person who is allergic to pollen grains, it means that there is a specific antigen within the pollen grain that is causing the allergy, and you want to create an antibody against it so that the person does not have an allergy anymore. So you're going to take the allergen, the one that is going to contain the antigen, and inject it to the rabbit. So once the antigen is going to enter into the body of the animal, it is going to get into the lymphoid system and then get accumulated either in the spleen or in the lymph node. So once the accumulation within the spleen or the lymph node occurs, the B cells are going to travel to the spleen, and this antigen for which we want an antibody is going to attach itself to the B cell, and this is occurring in the spleen. So this spleen from the infected animal is removed, and we are going to take the cells out of the spleen. So we are specifically targeting the B cells. So that is the only cell that we need from the spleen, because the B cell has gotten activated in the spleen because it has attached itself to the antigen. So once the cells are removed, the spleen is removed, the cells are mixed with myeloma cells. Myeloma cells are nothing but cancer cells. Cancer cells are used in this case because cancer cells are immortal cells, meaning that they don't have a specific lifespan. Any other cell in any of the mammals that you take or the human body, all of them have a specific lifespan. They live for a specific period of time and then they need to die. But myeloma cells, cancer cells, don't. They do not die. They are immortal cells and they live forever. That is the reason why myeloma cells are used in case of the hybridoma technique, because once you have a hybridized myeloma cell, you can keep it forever, grow it how many ever times you want, and always get the antibody that you want out of that hybridized cell. Okay, that is the reason why myeloma cells are used. Another thing to remember in case of myeloma cells is that they are lacking the HGPRT gene, which we will come to later on. Now the cells are mixed with the myeloma cells using PEG, which is nothing but polyethylene glycol. It is going to help with the fusion of the plasma membrane so that the B cell and the myeloma cell kind of fuse together and become one big cell. Okay, that is the use of polyethylene glycol. Once the fusion with the polyethylene glycol has occurred, these few cells are going to be grown on something known as the HAT medium. HAT medium is a selective medium. So in any molecular biology technique, you always need a selective marker. So usually for plasmids, we would be using things like antibiotic resistance genes. So ampicillin, kanamycin, tetracycline resistance genes are there within the plasmid for us to select whether or not the plasmid has entered into the cell. The same can be done with LAC-Z gene, wherein beta-galactosidase is going to be active if the gene has not entered into the plasmid. If the gene has entered into the plasmid, the gene is not going to, the LAC-Z gene is going to be inactive. The same kind of thing will be used in this case. HAT medium is a selective medium. HAT medium is called so because it stands for hypoxanthine, aminopterin and thymidine. Now in this, there are two things that you will need to pay attention to. That is, one is aminopterin. Aminopterin is going to stop de novo purine synthesis. Now purines are two things, adenine and guanine. Right, so the body can synthesize adenine and guanine by itself, and that is known as the de novo synthesis, meaning that the synthesis is going to occur within the body's cells. Then that kind of a synthesis is known as the de novo synthesis. Now aminopterin is going to stop the de novo synthesis of purines, meaning that cells cannot synthesize purine by themselves. If a cell cannot synthesize purines, which is adenine and guanine in this case, then DNA synthesis is stopped. If DNA synthesis is not occurring, then a cell cannot divide, right? So purines are extremely necessary for DNA, also for RNA, but if purines are not getting synthesized, then DNA is not getting synthesized, RNA is also not getting synthesized, and the cell will eventually die because it cannot divide, it cannot grow. The thing with hypoxanthine is that is where the HGPRT gene comes in. HGPRT stands for hypoxanthine guanine phosphoribosyl transferase. This is an enzyme which takes hypoxanthine and converts it to guanine. Okay, so if hypoxanthine is present in the medium, then using this enzyme, hypoxanthine can be converted into guanine only if the HGPRT gene is active in those cells. Now HGPRT gene is not active in myeloma cells, but it is active in normal B cells. Okay, so regular B cells have HGPRT gene activity, but myeloma cells do not have HGPRT gene activity. So if you leave myeloma cells in HAT medium, they all die, because myeloma cells don't have the HGPRT gene. They use the de novo synthesis for the purines. They use de novo purine synthesis. But because aminopterin is present in the HAT medium, if there is a myeloma cell put into HAT medium, aminopterin is going to stop the de novo purine synthesis. So this myeloma cell cannot synthesize purines by itself. It can, however, make use of hypoxanthine to synthesize guanine. But because it does not have HGPRT gene activity within itself, it cannot do this function. So myeloma cells are going to die with HAT medium. If they are grown on HAT medium, myeloma cells will die unless they are fused with B cells. If myeloma cells are fused with B cells, although aminopterin is going to stop de novo purine synthesis, the B cells have active HGPRT. HGPRT is active in case of B cells. So hypoxanthine guanine phosphoribosyl transferase can convert the hypoxanthine to guanine, and that will then you have the purines that are necessary for either RNA synthesis or for DNA synthesis. So the HAT medium is a selective medium in this way, that if there is a single myeloma cell, it is going to die off because it does not have the HGPRT gene. Only if B cells are fused with myeloma cells, then they can grow, because B cells have the HGPRT gene activity, and then you have purines being synthesized because of the HGPRT gene activity, and you will have DNA and RNA being synthesized and grow. So that is how selection occurs. Only few cells will grow, meaning B cell, if it is fused with the myeloma cell becoming one big cell, only those cells will grow. If it is a single myeloma cell, it will die. If it is a single B cell, that is also going to die. Okay, so once you have the selection, only the fused hybridoma are going to grow. So the fusion between the B cell and the myeloma cell is termed as a hybridoma. So only these hybridoma cells are going to grow. So once you have hybridoma cells growing, you're going to isolate out the hybridoma cells and then dilute them. So you're going to mix them with a lot of growth media and dilute it so that you will have one single cell growing in each microvell. So you're going to tighten it out, pipette it out the media in such a way that each well in a 96 well plate is going to contain just one single cell. So you're going to grow a colony of cells using just that one single cell, and that is where the monoclonal comes from. So you're going to dilute out the hybridoma, pipette it out into a 96 well plate so that each well is going to contain only one cell, and you're going to grow a colony of cells from that one single cell, and then you're going to test out how much affinity does the antigen have for the antibody. So how much affinity is there for between the antigen and the antibody. So you're going to test it out. Affinity is basically the degree to which both of them can attach to each other. So the antibody and the antigen, to what degree, with what affinity they can attach to each other, that is tested, and the stability of the antibody is tested. How stable is it? Is it going to get degraded if it is stored for a specific amount of time? All of these things are tested. Then the antibody which has the highest affinity for the antigen and the highest stability is going to be selected, and those clonal cells, single clonal cells which gave rise to those antibodies, are going to be grown and regrown whenever we need the antibody. So this is how monoclonal antibodies are going to be synthesized.